Thank you very much, everybody, for staying till the very end for the last talk of the day. This is mostly my student named Dai's work, but the first two authors have visa problems. The third author, Wei Yu, is also in the audience, my undergrad student who's now a PhD student at USC. So this talk is about designing stable mRNAs. And for the first half, I will be talking more about background, like linear design, and the second half will be extending it to ensemble design. So as we all know, mRNA vaccines have been very successful, but there's a problem that they're rather unstable because RNA molecules are largely single-stranded. And because of the single-stranded, they're likely to degrade in your cell or even before entering cell. So as a result, you got less like spike protein or whatever antigens expressed. And as a further result, you got less antibodies induced. So our question is, how do you design more stable mRNAs using algorithms computationally, which will also be more efficient and express more proteins? So here's the mathematical problem. So I guess for this audience, I can be very fast because the translation process from mRNA to protein is deterministic, but the inverse problem is non-deterministic due to the redundancy in the genetic code. Each amino acid can be translated by multiple triplet codons, and if you just multiply the number of choices together, you got a humongous number because of a combinatorial explosion. So among all these, 10 to the power 600 something alternatives, which all translate to the same spike protein, which sequence should you use for vaccine development? Well, luckily, there was a paper from Moderna which suggested that you should choose the most stable mRNA for vaccine because they will stay the longest in your cell and they will produce the most amount of proteins. But the real question is how? How do you search in this vast ocean of alternatives the most stable sequence? So in my past work, I had this linear design algorithm, which is a very simple dynamic programming algorithm because to solve this problem, because here we want to find the most stable sequence in terms of the MFE stability. And MFE is another minimization among all the possible structures. So this is really a minimization over a minimization. So that's why you can apply a very efficient dynamic programming algorithms. So for example, via Lattice parsing that we published in Nature. And here I'll just give you a brief overview of the linear design, and then we will extend it to ensemble design. So the first important thing is the representation, which we borrowed the concept of word lattice, which is basically a finite state automaton or kind of DFA in computer science. So we kind of packed the choices of codons into a little lattice. It's often just a third letter that has a choice, but sometimes you have a leucine which has 6 choices, or serine and arginine. It's more complicated like this. And then you stitch all these little DFAs or lattices together. You got a very long mRNA design lattice, which encodes all the exponentially many different sequences from, every path from the beginning to the end is a valid sequence for vaccine development. But the big question is, how do you find the most stable one? So this representation does not solve the question, but it's a change to a very nice compact representation, which enables us to apply the most important second algorithm, which is also borrowed from AI in lattice parsing. So in the past, when we do lattice parsing, which is from NLP, my original field, we have a word lattice. For example, maybe I cannot hear very clearly whether you said meal or veal in isolation of context, but given like a language model or a grammar, you can disambiguate that the most likely sequence is actually I like this meal along with its syntactic structure. So this algorithm actually dates back to 1961, more than 60 years from today. I just applied the same algorithm to a different problem in COVID. That is, I keep the algorithm, but I changed the ingredients from English word lattice to mRNA design lattice, which encodes all the different design choices. And then instead of English grammar, which encodes grammaticality, I used the RNA folding grammar, which encodes stability. And same algorithm, which can find the most stable sequence in this huge ocean of exponentially many alternatives in about 11 minutes, along with its secondary structure, optimal secondary structure, the MFE structure. So that was all very nice, and I will not go into any more details, but just to say that the COVID experiments were very, very surprising. So we got more than 100-fold increase in antibody response compared to standard technique, which is code optimization used by everybody else. So this is published in Nature with the Nature News. But today I'm going to extend it to something more interesting, which is ensemble design. So what's the difference between linear design and ensemble design? Well, as you all know, RNAs are much more flexible than proteins. They fold into many different structures, millions of alternative structures in equilibrium. So they always switch around between states. And also mRNA needs to be very flexible because during translation, as ribosome goes through it, the mRNA has to constantly unfold and refold, unfold and refold. So if it's rigid as rock, it's useless. So rather than optimizing for single MFE structure stability, we want to optimize for the ensemble stability, which sums up all the other alternative structures, the whole Boltzmann ensemble. And we would want to prefer a flatter energy landscape so that you can be more flexible, stable and flexible. So that's the new objective called EFE instead of MFE. MFE is very easy because it's a minimization over minimization. That's why dynamic programming is easy. But EFE is a minimization over a logarithm over a summation. This summation is the partition function, which sums over all the alternative structures in the Boltzmann summation, like a softmax, which is very hard to do. So this new objective is likely NP hard. And how can we do? So we said, we cannot solve that question. So we instead use a continuous optimization technique, which is common in today's machine learning. So what we do is to define a probabilistic distribution over all possible designs. And instead, we optimize the expected value of the EFE, of the ensemble free energy, so expected EFE, and hope over the whole distribution. So hopefully this distribution will shrink in entropy, and it will be more and more certain and converge to like one hot distribution. So here's a bit more details. So you can define the distribution over lattices very easily, because for every branching node, you can define the distribution, which corresponds to these in the sequence logos. And then, so you just optimize the, then you just do the product and weighted sum of local distributions, and you got a whole sequence distribution. And then you can define the expected ensemble free energy and optimize it by gradient descent. I will not tell you any more details, but just so that the distribution will eventually shrink in entropy, and they will be more and more certain. You can see this kind of demo, and then eventually it will be more like a one hot distribution, which you can extract the sequence. So unfortunately, even this ensemble, even this expected EFE is hard to optimize because there's a summation, there's expectation over a logarithm over a summation. So we first swap the summation, the expectation and logarithm using the Yang inequality, so this approximation. But after this swap, we got a very nice objective because this is expectation, which is weighted sum, and the QX, the partition function, is a summation over all possible structures. So this becomes a summation over a summation. Now it's very much like a minimization over minimization. So mean over mean, or max over max, or sum over sum, you can optimize, but you can just not mix them up, like not mean over sum or sum over mean. And then this weighted sum can be, so we need to extend linear design in two different ways. The first extension is for the inner sum. Inner sum is relatively easy over all structures. That's the partition function idea. So you just basically replace the linear fold in linear design by linear partition. So it's a partition function version instead of just MFE structure. The outer sum is relatively novel. It's a weighted sum over all sequences in the distribution. So we just need a weighted version of linear design. That's probabilistic lattice parsing. So the previous lattice parsing, everything is uniform, but now we have a probabilistic distribution. So it's just probabilistic lattice parsing. Okay, I will not go into any details, just to show you the results. So we want to optimize delta G ensemble, so that is the ensemble free energy to be lower, the better. So it can effectively find sequences with lower ensemble free energy, and the advantage is bigger on longer sequences over the baseline of random walk, especially on very long sequence of spike protein, which has over 1,000 amino acids. And we also look at, for example, the spike protein of SARS-CoV-2. The x-axis is MFE, the y-axis EFE. It achieves better EFE, but with the price of only slightly worse MFE, because you have to sacrifice MFE a little bit, because these two are not, these two objectives are very much in sync, very much correlated, but at the end of the day, you have to sacrifice MFE a little bit, but you got much better EFE. And Interestingly, so you also have a lot of byproducts of optimizing EFE, ensemble free energy. The byproducts we observe are, first, you got flatter energy landscape that we motivated in the very beginning. So for example, for this protein, linear design is this ensemble, this energy landscape, and ensemble design is much flatter. And then it has higher positional entropy. That means more structural flexibility. Positional entropy is like, for this nucleotide, which nucleotide you would pair to. So that's a distribution, and that's the positional entropy. So we got a much higher positional entropy, so it's more flexible. And then finally, we also got lower average unpaired probability, or AUP. And AUP, sorry, I forgot a citation here. It's actually Rito Das's work that defined AUP, and they showed that AUP is related to RNA degradation. If you have lower AUP, it's less likely to degrade. So you can see we lowered the AUP a little bit over linear design. Okay, so with that, I would like to thank you. And my student Wei Yu Tang, who's in the audience, is now a PhD student at USC. He has another poster that he's presenting today and tomorrow on sampling design for a different topic, which is RNA design for different secondary structure. Yeah, but stay tuned, and you can go to his poster as well. Thank you. Oh, sorry, we used too little amount of time. Too fast. Good. Yeah, sorry. Yeah.
Very, very nice talk.
Thank you.
So I was actually wondering, because ribosome is continuously unwinding the
Yeah,
whole coding region.
That's right.
So then theoretically is if it's continuously with a high translation efficiency,
Yeah.
you kind of actually create a very unbased situation. And then unbased situation will land to high Shannon entropy.
Yeah.
So basically is then if theoretically that's the case, but in the real life, because every time when you unfold a structure, it's quickly refolded back.
Yeah, just.
So that means when you have an ensemble structure, when you actually lead to like an unpair,
Yeah.
then you actually also can think how much the window size will actually provide a relatively back, like a stable status.
Right.
So in other way is because eventually you still want to create a relatively stable refolding structure.
Right.
In that way, right?
So I guess you want to say, like, if I need to unfold, which other nucleotide am I paired with, right? If I'm paired with something very far away, it's probably a bit harder for me to unfold. If I'm paired with something local, either upstream or downstream, it's probably easier to do the local unfolding and the refolding.
So that's why I was wondering whether you can scan as a window size.
Yeah.
Where you actually estimation ensemble, then try to create these space when ribosome maybe still can have a balance rather than you create a huge high Shannon entropy situation.
Yeah, that's a very, very good point. We'll talk offline. Yeah, thank you.
This probably reflects my ignorance more than anything, but are we taking for granted that the lowest energy would still, in practice, maybe this is what the last question was in regards to, have a decreased efficiency at its function?
Sorry,
so like, the assumption is the lowest energy state would be the most stable.
Yeah.
But is it possible that there's a degree of trade-off with functionality? With
functionality, for example, translation efficiency. Yeah, for sure. That unfortunately, there's no slide for that. That's more explored in my previous work, linear design, where we actually optimize two objectives like simultaneously. The x-axis is the MFE stability. The y-axis is the second objective of codon optimality, which is measuring translation efficiency. We have a joint objective between the two objectives. So in practice, there is a lambda hyperparameter to control how much you want to have the weight on the second objective. Yeah, so in reality, it's not just optimizing for stability. It's a balanced optimization between stability and translation efficiency.
And then have there been or are there in the works structured means to experimentally verify this? Or is that something that labs have done on a case-by-case basis whenever they just want to show or validate their method?
Yeah, so in reality, for example, in the linear design paper, we tested about 7 sequences over the map. Some sequences are more stable, some sequences are less stable, but more translation efficient. So they mostly perform very well. Yeah, so we have a control variable approach that we saw that for the same stability, the higher efficiency is better. For the same codon optimality, then the more stable sequence is better. Yeah, but you have to test a few sequences to know. Yeah.
Fascinating work. Thank you.
Thank you.
Within this framework, would it be possible to add information about which positions you should add RNA modifications to?
Oh, like chemical modification?
Yeah, like pseudo-uridylation or something like that.
Yeah, that's a very good point. But we don't have publicly available energy model for, say, pseudouridine or meso-1 pseudouridine. And also, in real vaccine development, they do 100% replacement. Like they replace all the iridians with pseudouridine or 1-meso-pseudouridine. They cannot say, oh, this particular I replace it with, because that would be too costly. They have to be a complete replacement, right? But if you're allowed to only replace a few of these nucleotides, you can probably be a lot more stable. Yeah, but this is probably not very practical as a product.
But in your case, then could you add pseudouridine and uridine?
Yeah. If we have a full energy model with pseudouridine and uridine together, we can decide which nucleotide I want it to be uridine, which nucleotide I want it to be pseudouridine. Yes, that will be the best. Yeah, thank you.
Any more questions? Okay, well
Thank
then
you.
let's
Thank you very much
again. And